All right, now back to chapter 5, sections 4 and 5. So we looked at carbs and lipids. Now we have proteins. Um, so before we look at the structure of proteins, let's look at some of the functions. They do a lot of important functions in living things. First, some of them are enzymes and help facilitate reactions. They're found as part of the immune system. The antibodies, these sort of bluish purple white things, those are proteins known as antibodies, which help to identify foreign objects so that they can be dealt with by other cells in the immune system. Um, they also form part of our immunity, our memory. Some are storage proteins um, in eggs and casein in milk protein. There are proteins that essentially are for energy storage. In membranes, we have not only the phospholipids and cholesterol, but we have proteins embedded in them that allow for the movement of materials in and out. Extremely important. We'll see that because of the hydrophilic and hydrophobic nature of the phospholipids, a lot of things can't move through that part of the membrane, so they need a pathway to get in and out, and these proteins form that pathway. Some have a hormonal function, these chemical signals that are sent all around the body to elicit some kind of response, um, insulin, for example. Other membrane proteins are embedded in the membrane, sort of on the surface of the membrane, and they are receptors to receive chemical signals and help initiate some reaction by that cell. Muscle, these contractile and motor proteins, so actin and myosin, we'll learn more about them, these protein fibers that pass one each other when muscles contract and relax. Obviously, you have a lot of that inside of us. Um, and sort of a variation, a structural protein, collagen, for example, is part of the connective system in our body that helps to hold things in place, connects, with connecting our bones to each other and muscle to bone and just holding organs and such into place. Um, all right, but now, the basic unit or monomer of a protein is known as an amino acid. And so here we see that amino group, the carboxyl group, that gives it a acidic property. There's a central carbon, a hydrogen, and what's known as the R group, or the variable group. And so, as you can see here, the amino group, carboxyl group, and hydrogen central carbon are all the same in each of these, but it's the yellow box here that is the R group, and the green box in this case. Now notice they're showing this a little different in that in solution, as we said, the amino group can gain a hydrogen and the carboxyl group can lose one. So they're just showing the ionic form of it. Um, so you have some of these R groups, these side chains. Again, here some of these that are hydrophobic and they don't have a charge. Others that are hydrophilic and have a charge. Um, some that are more on the acidic side, some that are more on the basic side. But these are the 20 amino acids, or at least there should be 20 there, I think, that we find in our proteins. And so in building proteins, which we'll talk about later, you just take various combinations of these amino acids and just bond them together through those dehydration synthesis reactions in these excuse me, long chains. And there we go. There's this long chain, one amino acid after another. Um, the protein polymers are known as polypeptides because the technical name for the bond that forms between amino acids are known as peptide bonds. So you just have a bunch of them, a polypeptide. All right, so proteins have several layers of structure. The primary layer of structure is just the strand of amino acids and the order in which they are in that protein. The secondary structure is, and the purple ribbon represents the primary structure, just the strand of amino acids. The secondary structure, in some parts of a polypeptide, you will get these things called alpha helices and beta pleated sheets where there's hydrogen bonding between amino acids that are close to each other. And so they can form these little curlicues or these sort of like these bent sheets. Think like the on a washboard, those old washboards that the I don't know bands use sometimes, or you rub the spoon against it. Sort of like that. It has that sort of I don't know convoluted shape to it. All right, tertiary structure is when you take the primary and secondary layers and then you fold it up even more. And that image, this image is on here twice, I'm not sure why, but so what you're doing with tertiary structure is you're having more distant amino acids bonding to each other. And it can happen again due to hydrogen bonding, disulfide bridges, ionic bonds between amino acids that have an opposite charge, and just those van der Waals interactions. So all these things can cause this more convoluted structure to form. Um, and so all proteins are going to have that level of structure. Some proteins have the fourth level of structure, and that's when you take different protein subunits and put them together. A good example of that, well, is this transurethrin one, and also hemoglobin. Hemoglobin is 4 separate subunits put together. But all proteins will have the tertiary level. This sort of tertiary and quaternary, this three-dimensional structure is very important for a protein because, for example, with enzymes, they'll have an active site, and let's just say, for example, this were the active site, and it has to have a particular shape so that molecules it's interacting with can fit in there. And so if that shape is not correct, it can cause that protein to not function properly. And so, and you get things like mutations, it can cause the protein structure, as is shown here, to change, and so then that protein doesn't, excuse me, function so well. So here's hemoglobin with its 4 subunits, and with sickle cell anemia, notice you have a difference in the amino acid sequence, that primary structure, which causes the secondary and tertiary structure to be different, and so this hemoglobin doesn't work as well and causes problems. As we'll see, the protein structure, the primary structure of the protein, is a result of DNA, and so when you get mutations in DNA, it can cause mutations in the protein. All right, this is brought up here, but we're going to cover this in much more detail later. As I just said, we use DNA as the template, if you will, to make proteins. RNA is made in the meantime. It's sort of the intermediate step. The RNA is made in the nucleus. It travels out, and it's used in the cytoplasm of the cell, particularly with ribosomes, to make the proteins. So these are the processes of what's called transcription, where we make RNA using DNA, and then translation, where we make proteins using the RNA. Again, we'll study that in more detail later. All right, last but not least, our fourth large organic molecule, important living things, nucleic acids, DNA and RNA. Okay, so here's our basic unit or monomer of the nucleotide. The monomer of a nucleic acid is the nucleotide, and so it consists of a sugar, a phosphate group, and a nitrogen base. In DNA, the sugar is known as deoxyribose, thus deoxyribonucleic acid. And in RNA, it's known as ribose, which is ribonucleic acid. So the sugars are somewhat different in DNA and RNA. The phosphate is the same all the time. And notice the phosphate and the sugar alternate, and they sort of make the side or backbone of the molecule. The nitrogen base sticks off the side, and there are 5 different types of nitrogen bases: cytosine, thymine, uracil, adenine, guanine. Notice adenine and guanine have 2 rings. They are known as the purines. And cytosine, thymine, and uracil have a single ring. They're known as the pyrimidines. the way I remember this is pie cut. Pyrimidines are cytosine, uracil, and thymine. And yeah, I know this is just blowing you away here. And the purines are POG. Purines are adenine and guanine. That's how I remember it. Picut and POG. Um, all right. So basically, in in DNA and RNA, you've got these nucleotides, and you've got 5 different ones because you've got 5 different nitrogen bases. So you'll have one with cytosine, one with thymine, uracil, adenine, and guanine. And so they come in a different order in our molecule, which makes DNA between us all different. Um, with DNA, you have 2 sides to the molecule, and the nitrogen bases in the middle are bonded to each other, which is what, through hydrogen bonds, which is what holds the 2 strands, 2 sides of the DNA molecule together. We'll see that for the most part, RNA is single-stranded, but like proteins, it forms this sort of 3-dimensional shape often, at least in the case with, say, transfer RNAs, which allows it to do its job. Um, we'll have chapters, there's a chapter later in the book that specifically focuses on DNA and then also the process by which we make RNA. And so we'll need to know more details there. For now, just know that DNA and RNA are examples, or are the nucleic acids we have. And there are some differences between them in terms of the sugar. Oh, and notice DNA has cytosine, thymine, adenine, and guanine, whereas in RNA, the uracil replaces thymine. So RNAs have cytosine, uracil, adenine, and guanine. Okay, I think that's it for chapter 5.